Bonjour.
Hello and welcome. Welcome to this very interesting interview with Marc ANTONINI, who is the Director of Research at CNRS, and he is working on the topic of the data storage based on DNA. And this is not just storage of DNA, but the storage through or on DNA. So this is an absolutely fascinating revolution in the IT as well as in the biology. And this is the innovation, a revolution that opens for us new opportunities for the storage of information by using the density and the solidness, the robustness of the DNA, by also revealing a very important technological challenge. So without further ado, Marc, thanks a lot for being here with us. Could you please explain to us the fundamental aspects of this DNA-based technology and its applications as well, and probably you can also share with us what you're doing in this field. Yes, thank you very much. Well, first of all, thank you for having invited me to this convention. Hello, everyone. So yes, indeed, the storage of the digital data on the DNA. This is a pluridisciplinary field, as you probably already understand, because here we're talking about chemists, but also biologists. We're also talking about the IT and biological IT. So this is the field which is very pluridisciplinary. And I would even say that the scheme in itself of the coding of the data is quite simple, meaning that we have to convert the binary data into the data as we call quaternary data, which is based on the DNA scheme. So basically, we're talking about this quaternary code. And once we have transformed this binary information, all the files that you have been using normally will be stored in the molecules of DNA, which will be a synthetic DNA molecules. And once this data is stored, it has very interesting specificities, because the DNA can be preserved for thousands or millions of years without any alterations if it is kept in a very favorable environment. Let's say right now I have brought this very small capsule. Probably you cannot see it because it's too far. But this is the capsule that has been created by Imagine, which is the French company. And inside of it, there is a DNA, which is a synthetic DNA, dehydrated and preserved in the oxygen. And right now we have approximately 500 to 2,000 years of preservation of this DNA. So we can basically keep it at the ambient temperature without any CO2 emissions for a very, very long period of time. Then also in this process, it's important for us to be able to get back this data from the DNA. And we're using here the classical solutions that we have here in biogene field, meaning that in this case, for this specific storage, we have to open the capsule and get the DNA from it and then read it and then have the PCR sequence. And after this sequence, we will be able to get this information, and this will be as a quaternary code. So we will be able to decode it and transform it into the binary information. So how does this methods of data storage differ in terms of advantages and challenges from the classical methods like hard drive and cloud service? Well, in terms of advantages, as I have already explained it to you, this is the solution that is a very, very long-term solution, the solution that is like forever. We can basically stock it for a very long term with basically zero energy consumption. And we can imagine that in this small capsule, we can have one or many data centers. So that's very compact as well. And we don't have any CO2 emissions. So this is very environment friendly. Now, in terms of challenges, I would even say like the advantage with respect to the hard drive or cloud service or magnetic field or something, we don't have to migrate data, because once it's stored in DNA, it's very stable and it's robust. So we basically have clones and copies of what has been stored, because we have many different capsules that will have this information, and the density is very important within each capsule. So we don't really have to do all of these migration processes that consume a lot of energy. and that are done all the time in the data centers in order to migrate the data from the hard drives to some other hard drives, because we have a certain lifespan for a hard drive, and it's of approximately 10 years, whereas for the magnetic band, we have a little bit more years. So all the time, we still have to do this copying process. And that's kind of a problem that is not existing with the storage on DNA-based technology. So we have talked about the storage technology and the sequencing as well. Yes, indeed, we are doing it in the lab today. So, of course, we have some startups that have been created, and they're being created permanently on this technology. There are also some big companies that are working on this aspect, and we also have some American companies that are doing it. So, in fact, the important challenge, well, we have several important challenges. First of all, in chemistry, it's important to accelerate the synthesis chemistry so that it will go even faster, and scale it so that it will be able to absorb the exponential increase of the data on the planet. And we're here basically interested by the archive data, the data for which we don't really have to have frequent access and quick access. Well, probably we will just store them and access them only once or twice in X years. So we are interested in this type of data, and we are trying to accelerate from the chemical point of view the synthesis, because today for 1 byte we have 100 seconds of time that are needed. So we have 10 kilobits per second for the writing. So it's approximately 10 gigabyte per second for a hard drive. So we can see that the difference here is quite important. So the project that I'm directing right now is called Molecular Archive, and it's financed by France 2030. And right now we are looking at this writing process, and the speed of it is coherent with the network. Well, it's quite good, but we actually have to improve here, because we're looking at 2030, 2035, and we would like for this technology to be a real competitor of the hard drive. And also there is this question of sequencing, because right now we are talking about the third generation sequencing, and this sequence is to 2 to 3 megabits per second. So we would like to scale all of this and go even faster, which is why we continue working on these aspects, and we're working on this sequencing. So what is your vision of the future for this technology? Can you probably explain us what are the major advances, the major progresses here, and how can you really imagine the future of this technology? Now, I really believe in this technology and in its future. I believe that within a couple of years we will have the data centers that will offer to us this cold archiving, at least at the very beginning, probably not in the capsules just like this, but probably in some other storage systems that will be a little bit more automatic. Well, this is something that I believe. I'm a firm believer of that. Well, of course, we still have some hurdles that we have to overcome. Well, I would say hurdles at the level of chemistry and molecular biology, but also we have to make sure that we can treat IT information and convert it in the needed format, and then we have to be able to get it after the sequencing, because we can imagine how the system works, and we understand that the decoding process must be done properly. And these are the challenges that are from the IT domain, but which is my domain. We also have this random access, and for example, if in this capsule we have one data center and we have several billions files, well, you will not be sequencing the whole data center to get just one file, because this will take too much time. So we have to create some hybrid solutions with IT and molecular structures, and like a hybrid between both of them, so that to be able to somehow extract the files much faster, and that will represent basically the brain of the thing that I would like to sequence here. So we still have some hurdles and some challenges that are here in front of us. Nonetheless, today, many people believe in it, many companies believe in it, and there is a huge movement in the United States toward this storage on DNA. In France as well, we are trying to do our best, so we have some projects here, and the projects are aiming at offering a solution here, because in France, I believe that we have all of the competencies from one end to the other for the storage, for the chain of storage, and Europe is also very interested by this chain of activity. Last year, there was this project that was launched that is called Pathfinder for the DNA-based storage technologies. There is another application that I would like to mention here. Well, this is also storage, the storage on the molecules, the storage of information, but for the marking and traceability, I would say that we could directly put and mark different products and, let's say, some substances as well. Well, for medicine, this will be drugs. So we can probably put the information on the boxes and put the information related to materials and so on. And in the molecules, we will be putting digital information. So we will be able to track materials. Well, this is very dense, actually. So I would say some nanograms of a molecule can contain gigabytes of data. So we actually can have a traceability here as well. So that's very interesting. And I would say that throughout the development of these technologies, especially for the medicine, for example, the application can be much faster applicable and much faster usable than the storage itself. Because here for us, the main slowing down point is especially the scaling. Because today we're working at megabytes, but if we would like to go to gigabytes or exabytes and so on and so forth, tetra, we are very much limited by time and by cost because, well, today we have to synthesize this DNA, whereas when we're talking about marking and tracing solutions, we don't really need to write many things. Now, the application of the DNA, the density of the DNA, this is something that can be used very quickly and easily here. Well, that's very interesting. This is futuristic technology. Yes, indeed. What is really interesting is that nature actually becomes this futuristic technology for us, and we're using it for the storage of IT. So it's not really IT that is serving biology, but right the opposite. And just to finish it, I would also like to address the ethical issues and some security concerns associated with the storage of data in DNA. So now, with the ethical concerns, well, this is not something that is living. So this is all synthetic. So it just represents the binary information that is encoded. So we don't have any coding sequences like the DNA or virus or whatever, so we don't have any genes inside, so we're very clear on these aspects. Now, in terms of the data safety, we can imagine that if we're looking at these small capsules, this is disconnected from internet, so we have absolutely no access to internet, so we can just take the physically the capsule in order to access this data. Now, we can also have some other systems that we are foreseeing for like 5 years later or 10 years later. This will be probably semi-automatic, and probably this will also be connected to the internet, and there will be some binary quaternary connection that will be automatized, and then we will have to be really careful and attentive to these aspects related to safety, and we will have to do it either through cryptography of all of this data in binary format or in quaternary format. We will have to see how it will happen, and then it will be probably done privately, and it's possible also to store the data somewhere and have a limited access to it, like physically store, I would like to say. So what I would like to say is that in theory today, if I wanted to have all the IT data of the planet, it will have to fit on approximately several hundreds of grams of the DNA, so it will be like a shoebox. Thank you very much. We still have a couple of minutes that are left. We are a little bit early, but thanks a lot for this information. This was very informative, very interesting, and hopefully I will receive additional information very soon from you. Yeah, this will be with pleasure. Hopefully this process will be quite quick. Thank you.